scientists uncover the bacteria behind the sea star decline in the Pacific Ocean. Over 10 years after a baffling illness began decimating billions of sea stars along the Pacific coast, researchers have finally pinpointed the bacteria responsible for this lethal disease. A collaborative effort involving at least 15 scientists from various organizations aimed to unravel the mystery behind the deaths of these marine creatures. Understanding the cause of this epidemic is crucial for initiating recovery efforts for both the sea stars and the ecosystems that have suffered due to their decline. After 4 years of extensive testing and detailed DNA analysis, the team discovered a specific bacterium consistently found in the 6 sea stars, which was absent in healthy specimens. This groundbreaking study brings renewed hope for the future of sea stars, potential treatment options, and the possibility of restoring the kelp forests that depend on these creatures to manage sea urchin populations. As noted by co-authors Melanie Prentice and Alyssa Gaiman from the Hakai Institute and the University of British Columbia, the findings will be published on August 4th in the journal Nature, Ecology, and Evolution. While many people refer to these star-shaped animals as starfish, scientists prefer the term sea stars because they are not fish. They belong to a broader group of marine animals known as echinoderms, which also includes sea cucumbers and sea urchins. What led to the sea star crisis? The first reported case of sea star disease emerged from a tide pool in Olympic National Park, Washington, in June 2013. The illness quickly spread, affecting sea stars in Sitka, Alaska, and along the coasts of British Columbia, San Diego, and even reaching as far south as Mexico. Infected sea stars exhibited lethargy, developed lesions, lost limbs, and disintegrated into mushy masses within days. According to the National Oceanic and Atmospheric Administration, NOAA, the pathogen was capable of wiping out a healthy group of sea stars in just 24 hours. As reported by USA Today in 2013, between 2013 and 2017, although they're the most vulnerable to the disease, it has been detected in more than 20 different sea star species. The vast kelp forests have also suffered due to the decline of sea stars, as these creatures play a vital role in controlling the population of sea urchins, which, if left unchecked, can devastate kelp ecosystems. What actions did the researchers take? In February 2020, just before the COVID-19 pandemic took hold globally, a large group of researchers convened to strategize on how to recover sunflower sea stars. As shared by Prentice and Gaiman in an interview with USA Today, their collaboration included the Nature Conservancy, the Tula Foundation, and researchers from the University of Washington and the U.S. Geological Survey. Between 2021 and 2024, the scientists collected wild sunflower sea stars from at least 6 locations in British Columbia and Washington, conducting a series of controlled experiments. Interestingly, while the scientists were working to halt the spread of a deadly virus in sea stars, the world was simultaneously grappling with the COVID-19 pandemic. As they quarantined the collected sea stars, team members were also isolating themselves to prevent the virus's spread. The mystery of sea stars unraveled. Did the researchers experience an aha moment? Indeed, Prentice recalls a day when she was analyzing microbial data from healthy sea stars and comparing it to data from those suffering from the disease. Just before a meeting with Gaiman and colleague Grace Crandall, she noticed a significant presence of Vibrio pathogens in the 6 sea stars. During their meeting, she shared her findings and they began analyzing the genetic sequences of the Vibrio they had identified. The culprit was confirmed to be Vibrio pectenicida. It was immediately clear that this bacterium was present in all of our diseased samples and absent in the healthy ones, Prentice stated. The team spent an additional year validating their findings, consistently gathering more evidence linking the pathogen to the disease. Understanding Vibrio. Vibrio is a genus that encompasses a wide variety of species found in marine environments, many of which are known to cause diseases. One type of Vibrio is notorious for killing oysters and can be fatal to humans, often referred to as flesh-eating bacteria. Another strain is responsible for cholera. Gaiman noted that the moment of discovery was particularly surprising because the team had anticipated that finding the answer would be complex. It turned out that the pattern was visually apparent, and we could see it clearly, she remarked. That was a surprising moment, and it made us realize we might be able to solve this issue. Kevin Lafferty, a marine disease ecologist and senior scientist with the U.S. Geological Survey, who was not part of the study, praised the team's efforts in a companion piece published in the journal. The research is truly remarkable and required immense dedication and hard work, Lafferty told USA Today. Diagnosing marine diseases can be challenging due to a lack of expertise in the field. This is a significant obstacle, and they overcame it through diligent effort and modern molecular techniques, he added. The ability to analyze a drop of seawater and identify 100 bacterial species is a monumental advancement in understanding microbial diseases, particularly in aquatic environments. What lies ahead for sea stars? This discovery opens a door that was previously closed to us because we lacked knowledge of the disease's cause, Gaiman explained. Many individuals are dedicated to saving this species, she continued. There is a wealth of work that can immediately benefit from our findings. Researchers are currently developing a diagnostic test similar to a COVID test, which could be crucial for assessing the health of sea stars and their environment when considering the transplantation of sea stars to aid in population recovery, according to Prentice. This will be immensely beneficial for our management strategies regarding the species. However, questions remain. As noted by Lafferty, the origin of the pathogen is still a mystery. Could it be transmitted from the mollusks that sea stars consume, similar to how humans become ill from eating infected oysters? Is it possible for the disease to spread from one sea star to another or through aquaculture practices? These inquiries and more will likely shape future research directions. Given that there are still healthy populations of sea stars in Alaska and British Columbia, as well as a few in Washington, researchers, armed with the knowledge of the disease's cause, hope to incorporate some of these healthy sea stars into captive breeding programs to cultivate individuals that can withstand the pathogen. Other avenues of exploration include identifying a probiotic that could bolster sea stars' defenses against disease, akin to methods used to pre-treat certain corals, or discovering a naturally occurring marine virus that specifically targets harmful bacteria, similar to the virus being utilized to aid in the recovery of abalone populations off the Southern California coast. We hope that one day we will be able to reintroduce sea stars back into the wild in areas where they have been lost, Prentice expressed. Contributing, Elizabeth Wise, USA Today.